if you wouldn't mind joining me to welcome Nic Dzamko. Thanks, Nic.
Thank you very much, Simon. It's a pleasure to be here today, and thank you for reminding me what I used to look like with hair. Can people hear me okay up the back? Yeah. Now I just need to work the slides. It's just the arrow keys. Okay, thank you. So I'm a basic scientist. I'm not a medical doctor. I'm hoping my presentation today won't be too scientific, but if you were sensible, you would have fueled up on coffee. And we'll see how we go. So we heard a lot of talk today already from Eli and from Professor Halliday about biomarkers. But what actually is that? Biomarker is just the short term for a biological marker, which in its very basic sense is basically something that we can measure in a person that tells us they are different from someone else. So in the case of Parkinson's, we want biomarkers that will tell us, what is different from someone who doesn't have Parkinson's. Now, lots of things can be used for biomarkers. They can be genomes or genes. They can be proteins. They can be lipids. They can be sugars on this slide. There's other things that are not on this slide. Imaging biomarkers, for example, are one obvious thing. But basically what we want to do is we want to use these biomarkers to tell us different things about Parkinson's. So, I don't know, let's see if this works. Okay. And so on one level, we can use a biomarker to actually kind of detect or even diagnose disease. So most people will have gone through a journey, of course, of being diagnosed with Parkinson's. It's not very quick. It's not very easy. You don't just turn up to a doctor, give some blood, and then find out in 2 weeks that you have Parkinson's or not. But that does happen for other diseases. And, we want to be able to get these diagnostic biomarkers. We want to be able to find them earlier and more accurate. Because, we've heard a lot of talk already about Parkinson's is actually quite complex. We want biomarkers in particular that can monitor Parkinson's or tell us about the disease progression. We want to be able to say, for example, and there was a question earlier this morning about, what is my risk of getting dementia, for example? Well, we don't know at the moment, but can we find biomarkers that might tell us if someone is diagnosed early? Can we find things that might predict the future course of their disease so we can actually start to tailor an individual therapy for them? And then, Parkinson's clinical, it's diagnosed clinically, and a lot of the clinical symptoms can overlap with other diseases. I had an auntie-in-law who was originally diagnosed as an alcoholic by her GP. And she was trying to tell her GP that she doesn't drink. And the GP was like, well, that's what all alcoholics say. Eventually, she went to see a neurologist because she kept getting kind of classical, movement disorder symptoms. The neurologist diagnosed her with Parkinson's. She was put on Parkinson's medication, but after a couple of years of not responding, they realized that she probably didn't have Parkinson's. And she was eventually diagnosed with a related disorder called multiple system atrophy. So, there's quite and if that was found earlier and treated earlier, then, it might have been a different outcome. So there's lots of things that we can use these biomarkers for. And we also heard from Professor Halliday, the way that the treatment is going for Parkinson's is, can we find biomarkers that will stratify people into the clinical trials that they need to go into? So if there are lots of different causes or lots of different ways to treat Parkinson's, how do we tell who gets which drug and who gets it when? And all of these things all get lumped into the generic title of biomarkers. So me and my research team in particular focus on this aspect of protein-based biomarkers. And more recently, we've started to journey into this world of lipid-based biomarkers. And that's what I will tell you a bit about to start with. So proteins are basically what you're made of. They do all of the functional work inside of your body. They are the enzymes that run all your metabolic processes and things like that. There's actually millions of proteins inside of your blood. No one knows exactly how many. That's based on computational modeling because we actually don't know them all. Most of them are yet to be discovered, and a lot of them because they aren't discovered, we still can't even measure them. But if people have gone through Simon's clinic and donated blood, what we do is we take that blood up to our lab and we start to measure proteins. Again, we're looking for the biomarkers that can tell us what might be different in someone's blood for someone who has Parkinson's disease compared to someone who doesn't. I'm not going to go through all of our work, but we do have a few biomarkers that we've worked on over the years. Scientists love to have little acronyms. We speak our own little language, but all of these abbreviations are different proteins that we've measured, that we've started to implement, as Simon suggested, or translate to biomarker assays that are used in clinic. And I guess the best example I can give you is some of our work around LRRK2. So LRRK2 is a protein. It was discovered in 2008 that if you have a mutation in your gene that makes this protein, that you're at a greater risk of Parkinson's than someone who doesn't. So this was discovered, but no one knew how to measure the LRRK2 protein. So we couldn't actually tell if it was different or not. So what we did is we spent a couple of years in the lab trying to develop ways to measure it. And it took a couple of years, but we actually did detect an assay that could measure the LRRK2 protein. So that was our biomarker. And then over the next 8 or so years, that biomarker gets tested in different clinical populations around the world. So other people kind of take your idea and they test it in their lab with their patients, and so on and so on and so on, until it builds up a big enough data set that people are convinced this biomarker actually works and is actually real. And this biomarker now is used in clinical trials. So over the course of almost a decade, we've gone from a discovery to something that's being used in clinical trials. And that's actually quite quick, to be honest. I think the LRRK2 story is one of the quickest, things from discovery to being implemented into a clinical trial. And that's being tested in the US and in Europe at the moment for people that have this specific mutation in their LRRK2 protein. So I also wanted to quickly touch on lipids. So there's hundreds of thousands of these lipid molecules in your blood as well. And the more things we measure is our philosophy, the more likely we are to detect these biomarkers or these differences. So we also started a project looking at lipids. Not many people have done this before. We're always trying to think of new things to look at and new things to measure. And what we were able to do is develop a mass spectrometry technique. This is kind of a really fancy machine. It can measure up to 1,000 lipids from a small blood sample at a time. And we did this for around 500 people. What these kind of graphs show, they're basically cluster plots. And the different colors are the different groups. So green, I guess I've got to read my screen here because I can't remember, but green and orange are people that don't have Parkinson's. So you can see green over here and orange over here. And then we have people with Parkinson's disease with and without LRRK2 mutation. And what the graph is showing is that you can really distinguish or differentiate these groups based on the lipid profiles of these people. So if you have Parkinson's, you'll sit over here and you'll look very different from someone who does not have Parkinson's. And that's quite exciting. This is kind of an early discovery phase of a biomarker, which we then want to try and measure and replicate. And then this has to go through these validation steps of seeing if it can be reproduced around the world in different cohorts of people. The other thing that we're really interested in doing is trying to predict the course of Parkinson's disease. We know that it's different for every person, and I don't know, I can't speak as someone with Parkinson's experience, but if you wanted to know what the future held for me, can we find biomarkers to answer your questions? So what we normally do here is we might collect a lot of information from you. If you come through Simon's clinic, for example, you may go through all of the clinical tests, then we might take some of your blood and we might start to measure our biomarker panels in it. And then we start to ask, these fancy machine learning algorithms that Professor Halliday mentioned earlier, can you use this information to predict what happened to the people in the future? So if you keep coming back to the clinic year after year after year after year, and we can build up the data set, we can start to see the patterns that people might fit into, and we can ask the machine to try and predict those patterns and tell us what are the biomarkers that may tell us if someone is going to get, worse quicker than someone else, for example. And that's what this graph is. It's a graph of a prediction model, and it's saying that people with higher inflammation may actually get worse a bit quicker than people who do not, in terms of their motor symptoms. Now, these models are not perfect. If this was a perfect model for people who like mathematics and statistics, all of these dots would lie along this straight line. So the more scattered the dots are from the straight line, the more inaccurate your model is. And that's because these are really data-hungry models. They like to have a lot of stuff put into them to try and find, what actually are the key biomarkers that can predict this disease progression. So that's another thing that we're working on now with our proteins and our lipids. So I had two parts to my talk, and that's kind of the end of the first part, which was on biomarker research. So that's what we do, and our biomarker research is continuing. We're trying to find and validate these novel biomarkers for Parkinson's disease, with our aims of really trying to help improve the timing, the speed, and the accuracy of a Parkinson's diagnosis. And in particular, can we help improve clinical trial outcomes, either by having biomarkers that can tell us objectively if someone is getting better or not when they're on a drug therapy, or can we have biomarkers that help us put patients into specific therapies that are more likely to work for them? And I just had the last point up there that biomarker research actually can't happen without the help of patients, without the biosamples that are donated to us. For example, for the people that come through Simon's clinic, the scientists like me have actually nothing to work with. So if you've donated blood, thank you very much. You've helped, advance Parkinson's research, and that's greatly appreciated. I also had the second part to the topic I was given, which was new drugs to treat Parkinson's disease. So this is a picture I stole off the internet. It's basically showing a drug development pipeline. And I don't know, people may be familiar with this or they may be not, but it's quite a process to get a drug all the way from a discovery to being used in the clinic. You can start off in the drug discovery phase, screening thousands and thousands of compounds to try and work out what might work and what might not for your particular drug target of interest. A handful of those might then go into what we call a preclinical model phase. So this is testing them in models before they're given to people to prove that they're safe, to try and build up the evidence that they actually will work and do what people think. And so this is kind of the space that I work in down here as a basic scientist. You can see down the bottom of this slide, the timeline that things might take as well. It's, 10 to 20 years to go through this process from a new drug to maybe licensed in a clinic. I mentioned the LRRK2 inhibitors before, about 10 years. They're actually here now in this phase, in phase 2 clinical trials in 10 years. And then you might take only a small amount of those compounds that come through the preclinical modeling might then go into the clinical trials, where you have your phase 1 safety trials and then your phase 2 trials to see if the drugs will work or not. And you can see that as they progress through the clinical trials, you need more and more people to try and show that they're working. That's not working. And you may end up with one drug at the end. So I wanted to talk about the preclinical models that we use in my team. And we use what we call these, or what people call these induced pluripotent stem cell models. So this is quite an amazing discovery. It was the 2012 Nobel Prize for Medicine. And basically what these people did, and I don't know, I'm a scientist, but I'm amazed sometimes by how people can come up with these ideas and discover these kinds of things. But before this discovery, it was kind of thought that a skin cell was a skin cell, a brain cell was a brain cell, a muscle cell was a muscle cell. They'd been made to do their job, and that's all they were ever going to do in their life. But what these discovered, and this was a Nobel Prize to a group in Japan and a group in the UK, they discovered that they could take those cells and they could put them back or reprogram them to these induced pluripotent stem cells. And induced pluripotent stem cells, it's basically how you start life. These are cells that can then be turned into any other kind of cell. So you can take someone's skin cell or someone's blood cell, you can reprogram it to an induced pluripotent stem cell, and then importantly for our neuroscience research, you can actually turn that induced pluripotent stem cell into someone's brain cells. So if you want to give me some skin or some blood, I can grow a dish of cells that will kind of resemble your own brain. And that's kind of new and that's kind of exciting because, you know, before this discovery, it was not actually really even possible to study human brain cells in a dish in this way. And that allows us now to, you know, we can't test new drugs on people. We have to know they're safe before we can give them to a patient, but we can treat your cells and we can kind of see how they might respond or react to different therapies. And of course, one of the bonuses of these cells is they actually make really nice pictures as well. So here's what they look like. These are induced pluripotent stem cells that have been differentiated into neural stem cells. This is what they look like under a high-powered microscope. We label them with different colors to label different proteins so we can start to measure the different kinds of things in these cells. You can take these neural stem cells and then you can turn them into these little networks of neurons. So this was once someone's skin that is now being turned into a dish of cells that kind of resembles their brain. And we can study these. We have developed now kind of high content or high throughput ways where we have powerful microscopes that can go through and they start to look at these dishes of cells and they try to work out what is going wrong in the cells from this patient. And then if we can measure something that is going wrong, then we have an assay where we can try use drugs to screen and stop that process from going wrong. And that's basically our preclinical models. I just have a couple of examples of this, this in action. So this is one study looking at the LRRK2 kinase inhibitors. We were part of the group that helped to build evidence that these drugs should go into a clinical trial. What this images show, they've been labeled green to show the protein alpha-synuclein. And we've already heard about alpha-synuclein today. It's the protein that can kind of clump in the brain cells. And then, I can't really see if this works. No. What you can see on this side are the cells as they look normally. So they're starting to accumulate this kind of green alpha-synuclein protein. That's right. And here we treated them with one drug, and here we treated them with another drug. I'm not sure how clear that is to people in the back of the room, but if you have good eyes, you might be able to see that there's less green signal when we treat the cells with our drugs. One of these drugs is from GlaxoSmithKline, another one was developed by Pfizer. And the graphs just are using the computer software to quantify the amount of green signal, and they're showing that the drugs are reducing the alpha-synuclein in these brain cells. Here's another example that we've published. Here we're using drugs that activate another protein called AMP kinase. So we start with our drugs. In this instance, we can give them a trigger to cause the alpha-synuclein pathology. So we can kind of make it look like the cells have Parkinson's disease. And then again, we can give our other two drugs. And hopefully you can see that the drugs again are reducing that green signal, the alpha-synuclein protein. AMPK is actually the molecular target for exercise. So if you're exercising, you're kind of naturally doing this rather than taking drugs. So we heard already about the benefits of exercise, and this is one of the ways that molecularly it actually works by activating this AMP kinase enzyme. A third and final example is work that we're doing with a San Diego biotech company called Neuropore Therapies. So we quite like it when people around the world want to come and use our models to test their drugs. In this instance, they're using something called a TLR2 inhibitor. So it's a drug that blocks a receptor that's thought to be involved in the uptake or the way the alpha-synuclein can aggregate in the brain. And again, following the similar pattern, the green is labeling the alpha-synuclein protein. We have some controls. Then we trigger our Parkinson's disease with our trigger to initiate the green protein. But again, here we treated the cells with the drug, and the drug clearly is reducing or even preventing the green signal from occurring. So that's the kind of thing we use these preclinical models for. They help to build the evidence. A model is a model. It's not an actual guarantee that this will ever work in a human patient, but they help to build the evidence that these drugs should enter a clinical trial if they prove safe. Obviously, we hope they'll be beneficial for patients in the future, even hopefully in the near future. And I just wanted to finish off that the other thing that we've started to do, we have another project going with a local Sydney biotechnology company. They're called Inventia Life Sciences. And what they've developed is this kind of cute little machine. This is actually a 3D bioprinter. So what we can do now is we can take those induced pluripotent stem cells, and instead of growing them flat in a dish, we can actually print them into 3D structures. And this is a picture of what we call a 3D organoid. And the thought is that a human brain is not a flat dish. It's actually a three-dimensional structure. Can we actually start to build things that look and act more like a proper human brain? Trying to make our models better and better and more reflective of what might actually happen in a person. I think that's all I have, and I have no idea if I'm sorry.